differentiation assay and walking through the steps that you need to know for how to do it. First thing you're going to need is a lab notebook. This way you can make an outline for whatever you're going to put in each individual well of whatever size well plate you're using, whether that's 6, 24, or 96. You're also going to need a falcon tube, a flask that has your cells in it, and then a collagen type 4 well plate. One last thing you're going to need is also differentiated media, either with NGF or without NGF, depending on what type of experiment you're doing and what you have in each well plate. Before you start working, you need to make sure that everything in the hood is sprayed down. I already sprayed it down before we started the video and went ahead and put the diff media and the well plate in there, but just as an example, spray it and put it in, spray it, put it in, and make sure your hands are extra clean before you go into the hood as well. Make sure you have the right size pipette for whatever you're taking up and out. So for this, we're taking up about 20 milliliters, so I'll have a 25 milliliter pipette. First step is you get the pipette out, making sure that the tip of it never touches the ground. You want to keep everything sterile. I usually loosen the lid up on whatever I'm taking fluid out of first, just to make it a little bit easier whenever I get to the actual step. Dispose of the covering. And now you can take your fluid out. The next step with your fluid that has your cells in it is to put it into a falcon tube. You're going to use the falcon tube so that you can spin the cells down and form a pellet down at the bottom once it's been centrifuged. One thing you can do while your cells are being centrifuged is to go ahead and make sure that everything that you don't need in the hood for the next set is taken out. That way nothing is too cluttered whenever you're trying to work. The first thing you need to do once your cells have been centrifuged is aspirate off all of the old media. Undo the cells. Make sure you can see where the cell pellet is. It's right here on this one. Tilt it and slowly aspirate off. Alright, you can leave just a little bit of media down at the bottom because you don't want to aspirate up your cell pellet and then you won't have any cells left. So for the next step, you're going to add media back onto your cell pellet. Some people will have you add dissociation buffer as well, and you'll do that first and wait a minute. We usually add 500 microliters for every 5 milliliters total, so you'd add 500 microliters of dissociation buffer and then 4.5 milliliters of media. But for these purposes, we're not going to worry about dissociation buffer. Some people might want to use it though to keep your cells from clumping together too much. So we will take out 5 milliliters of media. And since we're doing a differentiation assay, we want to use differentiated media, not complete media. And get that up to 5 milliliters. And then we're going to resuspend our cells in this new media. So gently put the media on, and then once you have all of the media into the T75 flask, just break up the cell pellet by pipetting up and down a few times. Don't go too fast because you don't want to get a lot of bubbles on there, but just take it slow and make sure that you have the pellet sufficiently broken up. So one thing you need to do before you decide to plate your cells is you need to count. That way you can get an accurate number of cells per well, which we're going to discuss later whenever we open up our 24 well plate. You need to take I usually do about 10 microliters and pipette it onto a hemocytometer. So here's the hemocytometer that you'll be using. The cover slip would go right there down at the bottom. Then you'll inject about 10 microliters of your cells, and through capillary action it will be taken up. Then you can count the cells under a microscope. Once you've counted your cells and you know what concentration you have, then you can start plating your cells. And you can open up your 24-well plate. For our experiments with PC12 cells, we normally use 3,000 cells per centimeter squared in our well. For a 24-well plate, there are 2 centimeters squared per each well. So that means 6,000 cells per well. So based off of your count here, you need to make sure that you do the math correctly to be able to add the right number of cells per each well. You'll also want to look back at your lab notebook and double check that you know what needs to go into each well, what treatment goes here, there, and all the way down for however many wells you're using. For our experiment today, we're just going to add some differentiated media without NGF to the wells, just as an example for how you need to add. The golden rule to go by is to add at least about half of the well up with media. That way the cells have plenty of food for whenever they decide to differentiate over the next few days. For this case, that means a little bit over a milliliter per each. I'm going to add about 1.25 milliliters to each of these. Once you have your wells all properly plated and ready to go, then you want to put it into an incubator. For PC12 cells, that incubator needs to be set at 37 degrees Celsius with 5% carbon dioxide being piped in. At about 3 to 4 days, you should start seeing differentiation in your cells if you've had NGF added. Now, when you're doing that, you want to make sure that you're taking pictures of the cells each and every day. That way you can see the differentiation develop over time. You don't want to only take it on day 3 or day 4 to see if there's differentiation, because then you'll have nothing to back up your results. I also like to write down in my lab notebook what I'm observing as I'm taking the pictures. That way later on, whenever you're looking back at the pictures, you have some words to go with it to back up what you're seeing. When you're taking pictures of your cells each and every day, you need to make sure that you get something that's representative of the well as a whole. So spend enough time to make sure that you're getting good pictures. I normally take a couple pictures that are at 10x magnitude. That way you can see the whole well and make sure it's representative of all the cells. And then look at 40x magnitude at the cells individually and see if I can see differentiation. Here's one example of cells with differentiation after about 6 days. That is what you're really looking for whenever your cells are differentiating. That's the goal. Okay, final step, once you've taken your pictures for your first day, open up the incubator, that again should be at 37 degrees and 5% CO2, and slide them in there. After the first day, you need to watch your cells every day and make sure that they don't need more media. Sometimes you're going to have to switch out the media, usually about every 2 to 3 days. That way the cells can stay fed and then they don't die during your experiment. Continue to take pictures, continue to switch out media, and you should be fine.